Howdy, Pima Aztecs. Welcome to Chapter 5, Microbial Metabolism. So this is going to be a whirlwind tour of metabolism. Metabolism is very complicated. It's way more complicated than what I'm going to show you in this video. I'll try to just bring out the important points so that you can understand the metabolism of microbes. Okay, so can anybody guess what molecule this is? There's 1, 2, 3 phosphates on it, and it's linked to A, the 4 nucleotides in DNA, adenine, guanine, cytosine, and does anyone know the next one? Thymine. Good. This molecule is ATP. So, adenosine triphosphate. This is a molecule that the cell uses for its currency. So, whenever it needs energy to build something, make something, it uses ATP as the currency. So, metabolism, like I said, is very complicated. This is a simplified. Oh, I just made up a new word. Simplified. This is a simplified diagram of metabolism. So inside this thing, so this is a cell. It may be getting ready to do cytokinesis. So it's pinching off the cell membrane from one mother cell to make 2 daughter cells. So metabolism in general, very simply. Here is a glucose molecule, C6H12O6, and then other nutrients here. So it could be micronutrients, macronutrients, but glucose is definitely a nutrient. Glucose goes into the cell, and it shows here that this process, catabolism, so catabolism is performed on the molecule glucose. So what that means is glucose is broken down, and when this molecule C6H12O6 is broken down, you can go through some serious magical biochemistry and make ATP, the energy currency of the cell. Anytime you do a reaction, some energy gets lost as heat. Can't not do that. But then, so ATP is made when glucose is broken down. Then ATP, that's energy, is used to build things up. That's called anabolism. So you're a cell, you take in glucose and other nutrients, you break down glucose, you make ATP, you use that ATP to build larger molecules. Well, actually it shows here in this cartoon, use ATP to make building blocks, and then those building blocks are put together to make macromolecules. So macromolecules would include things like nucleic acids, protein, carbohydrate, fat, and then from those macromolecules you use those guys to make your cell structures. So for example, your cell membrane, remember that's a phospholipid bilayer, so you'd need to make a phospholipid bilayer, you'd need some lipids. The macromolecules are also used for general cell processes like cell growth, cell division. And then you can also store the energy in those macromolecules in the form of carbohydrates and fat. And you guys probably know that fat is, lipids or fat in your body is an amazing storage form of energy. So a generalized definition of metabolism is that it is a collection of controlled biochemical reactions. So they're not just biochemical reactions happening willy-nilly. They are controlled, and they happen in cells. There's 2 major classes of metabolic pathways, and the 2 major classes are the anabolic class, that's when you build things up, and the catabolic class, that's when you break things down inside a cell. So, and in our microbiology world, it's important to note that some reactions will extract energy from nutrients, which is what we do as eukaryotes. Other reactions will extract energy from light. So that could happen in the prokaryotic world. It could happen in the eukaryotic world too. So in order to understand how reactions are controlled in the body, that means that you need an enzyme to bind to a substrate and make a product. And that's what this cartoon shows. So here's your enzyme, this purple thing. And here in this cartoon, it's got two substrates, this yellow thing and this blue thing. And the yellow thing and blue thing will fit into their active sites. So this yellow thing will go here, the blue thing will go here, and they fit, the substrates have to fit into the active site of an enzyme. And here it shows they are fitting in. So here's the yellow thing, here's the blue thing fits in. When the substrate, so that's the yellow thing and the blue thing, those are substrates. When they fit into the active site of an enzyme, you call it an enzyme substrate complex. And then the enzyme does biochemical magic on the substrates. In this case, it links them together right here. And then the enzyme releases something called a product. So you link the, kind of looks like a screw and a triangle, you link them together and you made a product. Okay, so we just talked about that. So why do you need enzymes? So if you are in a non-living organism, so if you're in the lab and you want to hook up these two substrates, what you could do is you could put the substrates in a solution, no enzyme, and you could jack up the temperature so high that there would be so many collisions between the yellow screw and the blue triangle that eventually you could form this product where they linked up. But usually in living organisms, you can't get that temperature high enough for the yellow screw and the blue triangle to collide and form a link. So that's why Mother Nature invented enzymes. And enzymes are catalysts, and a catalyst is just something that can make a reaction happen. And there's protein catalysts and non-protein catalysts. Most of the enzymes we'll talk about are protein catalysts, so therefore they're protein enzymes. So you need a catalyst to make the reaction run. This is the reaction. Here's the definition of a reaction from this cartoon. The two substrates bind to an enzyme and the enzyme releases a product. That is the reaction. Organic catalysts are called enzymes and they're usually protein. So remember the definition, if you are anything organic, you have to have a carbon in it. And really the truth is you have to have a carbon and a hydrogen if you are an organic molecule. In this case, an organic catalyst. If this cartoon were a catabolic reaction, then the enzyme would destabilize to break the bond. So if this were a catabolic reaction, the reaction would be going this way, from left to right. In an anabolic reaction, that's what is shown in this cartoon. Enzymes make sure that the yellow screw and the purple triangle collide and form a bond. Enzymes, you probably already know this, most of the time, and the name of an enzyme ends in ACE. And often enzymes are named after the substrate and or the reactant. There's 6 basic categories of enzymes. Hydrolases, so hydro, that's water, they break down molecules with the addition of water. So if you are an enzyme and you are a hydrolase, you use a water molecule to break the bond between your two molecules of interest. If you are a lyase, then you can break down molecules without using water. If you are a ligase or a polymerase, you are building up reaction or an anabolic reaction, and you join two molecules together. So for example, there are DNA polymerases and RNA polymerases, and they join the DNA and RNA molecules together. These guys are kind of cool. Isomerases. They do both anabolic and catabolic reactions, and all they do is they rearrange the atoms in a molecule. They don't add anything, they don't take anything away, they just take the atoms within a molecule and switch them to different sites. That's an isomerase. Very cool, Mother Nature. Oxidoreductases, those could be anabolic or catabolic reactions. They remove or add an electron. So oxidoreductases are used in redox reactions. And then transferases transfer functional groups like a hydroxyl group, OH, or a methyl group like CH3. They just transfer functional groups from molecule to molecule. And this cartoon is actually a protein with an active site binding a substrate. So this is a cartoon of just one pathway that we're going to study, not in this detail, so you don't have to know all this stuff, but it's cool because it shows the enzymes in glycolysis. So remember, previously in this video, I told you that cells take in this molecule, glucose, C6H12O6, and eventually they make, in glycolysis anyway, in this pathway, glucose is broken down to make an ATP here and an ATP here. So, but in glycolysis, in this cartoon, because the substrates are on the left of the arrow and the products are on the right, in glycolysis you have to invest a molecule of ATP here, you have to invest a molecule of ATP here, but then you can actually produce molecules of ATP in glycolysis. So, does it show it in this cartoon? You invest 2 ATPs, but you make 4 ATPs, so that means you net 2 ATPs in glycolysis. So here's the glucose molecule. The first enzyme, hexokinase, to act on the molecule glucose puts a phosphate on the glucose molecule. And now that molecule is called glucose 6-phosphate. Then the next enzyme to act on glucose 6-phosphate is phosphoglucose isomerase. So remember, isomerases don't add any atoms, they just rearrange the atoms that are already there. Anyway, the point of this cartoon is not to overwhelm you, but I just wanted to show you all the enzymes. So there's a phosphofructokinase, there's an aldolase, triose phosphate isomerase, glyceraldehyde 3-phosphate, etc. So if you want to do a reaction, a controlled reaction in the cells in your body, you need enzymes. Okay, so let's look at glycolysis in more detail. Carbohydrate catabolism. So remember, glucose is a carbohydrate, C6H12O6. So it's right here. Here's just a stick drawing of it, and then here's like a molecular model drawing of glucose. And then here's the pathways that we're going to study in metabolism. So everything in this green box is labeled respiration. Everything in this orange box is fermentation. And then you can see here glycolysis, which we just looked at that diagram of all the enzymes in glycolysis. In this cartoon, it just shows arrows. It doesn't show any of the enzymes, it doesn't show any of the molecules, it's just a real simplified diagram of glucose. Glucose undergoing the process of glycolysis. So in this cartoon, you can see that glycolysis is a participant in respiration, and glycolysis can also be a participant in fermentation. So anyway, you start as a C6H12O6 as a glucose molecule. You go through glycolysis, and the products of glycolysis, you get 2-pyruvic acids, and then you can see that those 2-pyruvic acids can either continue on here in respiration, or those 2-pyruvic acids can go to further processing in fermentation. And you can also see some more products of glycolysis are NADH. They carry electrons in the form of hydrogen atoms or ions to the electron transport chain. And here's ATP. So you get an ATP and an NADH, actually two. If molecular oxygen is present, then the 2-pyruvic acids can go to this linker step, acetyl-CoA, then they can go into the Krebs cycle where they can make more ATP. And then the Krebs cycle also produces NADH and FADH2, and those, including the NADHs from glycolysis, are carrying electrons to the electron transport chain, and then through a bunch of cool biochemistry, you can make even more ATP. So if you are a glucose molecule and there is oxygen present, and you get broken down all the way through such that your atoms go to the electron transport chain, you can make a whole bunch of ATP. Depends on the organism, but could be 32, 34, or 36 ATP. And glucose is being broken down, so that's called catabolization. And its primary source of energy for ATP, we talked about that. You can also use other sugars like fructose, you can use amino acids, fats. You can also use those to make ATP, but oftentimes those molecules are first converted into glucose and then glucose is broken down. But we won't go into those reactions. So both respiration and fermentation begin with glycolysis. So remember all this kind of green part of the cartoon is respiration, the orange part is fermentation. Glucose, C6H12O6, can be catabolized or broken down by the process of cellular respiration. That's when you completely break down the glucose molecule, C6H12O6, to carbon dioxide, which is what you breathe out, and molecular water. And then you also from that you make ATP. And we said this, when oxygen is present, so molecular oxygen, that's O2, molecular oxygen is present, you can go all the way to the electron transport chain, make a boatload of ATP. If oxygen is not present, glucose can also be broken down by this process of fermentation. So whether oxygen is present or not, glucose is going to break down to 2-pyruvic acids. If oxygen is not present, pyruvic acid is going to go over here into the fermentation process, where you get partial breakdown of the remainder of that glucose molecule, which is pyruvic acid. And what happens is the cells that are doing this, you get organic waste like ethanol in yeast. But then for us, we would use fermentation, and our breakdown product would be lactic acid. Okay. So if you're doing fermentation, there's no oxygen. That means you don't go through the Krebs cycle, you don't go through electron transport, and you get really low levels of ATP. But at least you can keep making ATP. Not very much, but you can keep making it. So throughout the adventure of metabolism, I like to do this. You don't necessarily have to do this for the exam, but I like to keep track of where my carbons went. I like to keep track of how many ATPs I get in each step. So for example, the net yield in glycolysis for ATP is 2. And also I keep track of my NADs that's funny, and FADs, because when they have hydrogen attached to them, they also are carrying electrons. And remember, if you have electrons, you're carrying electrons, and you carry them to the electron transport chain, which would be right here in our diagram. That's where you could get a buttload of ATP, which is what you want. You want to make a lot of currency, right? So, and in the cell, energy currency is ATP. So this is a simplified diagram from your book, and it's simplified because remember, glucose 6C6H12O6. Look here, it's it's only showing in the cartoon the 6 carbons. And if you notice over here, it doesn't name the enzymes that are involved in each step. But it does show you, you take one 6-carbon glucose molecule and you invest in ATP. That's called the energy investment stage. Glucose gets phosphorylated by ATP to form this molecule, glucose 6-phosphate. So see, it's got this phosphate on it because it took one of the phosphates, 3 phosphates from ATP, took one of those phosphates and made ADP, adenosine diphosphate. That's 2 phosphates. Okay, and then at this second stage, you also have to invest some energy to get glucose going. You invest another molecule of ATP, and now look what, and you use an isomerase, and then that's an enzyme. Then you make this molecule, fructose 1,6-bisphosphate. Bis is 2. It's got 2 phosphates on it. And then you can see the glycolysis pathway splits, and that fructose 1,6-bisphosphate results in, let's see, stage 4, it says fructose 1,6-bisphosphate is cleaved to form G3P and DHAP. And then DHAP is rearranged to make another G3P. Now this is interesting. Okay, Mother Nature, why don't you just, if you're going to make 3GP, why don't you just make 2,3-GP at once? Well, she didn't. She made DHAP, and then it gets turned into G3P. But check this out. This is really important to note if you want to understand this process. Remember, glucose started as a 6-carbon molecule. Look, you still have all those carbon molecules from glucose. But now you've got, because you've got 3 carbons in G3P, and then you have another 3 carbons in this G3P. And now look what happens. On step 6, this is the energy conserving stage. So you insert an NAD plus, and what happens is NAD plus gets reduced to NADH. Now this NADH got reduced, so that means it got an electron stuck on it with this hydrogen ion. So that means now you've got this electron carrier. And remember, if you can carry electrons to the electron transport chain over here, you can make a buttload of ATP. So then anyway, now you've got these 2 molecules, these 2 3-carbon molecules, and now you can start making ATP. So look at this. You bring in an ADP, and it gets phosphorylated, so it gets phosphate stuck on it, and now you make 2 ATP. So think about it. You invested 1 ATP up here, 1 ATP here, but now you've made 2 ATP. So you're still even. You haven't netted any ATP yet, but you continue down the process of glycolysis and you make 2 more ATP. So that means you have a net of 2 ATP from glycolysis. And you still have your 3 carbons. But one other product that you made is this 2 NADHs. That's 2 NADHs. So you're carrying 2 electrons to the electron transport chain, where you get a buttload of ATP. So glycolysis converts glucose into a product called pyruvate or pyruvic acid. That's these 2 guys here. In a bacterial cell, which is what we're interested in microbiology, glycolysis, so over here on the table, glycolysis precedes fermentation, which uses 1 glucose molecule, so we talked about that, you start with 1 glucose molecule, to produce a net gain of 2 ATP molecules. Don't know why I have that question mark there. That shouldn't be there. Okay, so we talked about glycolysis, that's this part right here, and you end up with 2 pyruvic acids, aka pyruvates. And then if there's no oxygen, you go over here to fermentation. If there's oxygen, you can continue here to this thing called the linker step. This linker step gets those 2 3-carbon pyruvic acid molecules arranged so that they can go into this guy right here, the Krebs cycle. And here's a cartoon or a drawing of pyruvate, aka pyruvic acid. And then here's the details of the linker step. So check it out. In the linker step, you give off a molecule of CO2 per pyruvate. So remember, there's 2 pyruvates. And you also, check it out, get an NADH, which is cool because remember, it carries electrons to the electron transport chain. And then you take acetate, here's a molecule of acetate and coenzyme A, and you make this molecule called acetyl-CoA. So it's a diagram of acetyl-CoA. That acetyl-CoA can go into this cycle, the Krebs cycle. So after glycolysis and before entering the Krebs cycle, so we're right here, this 3-carbon pyruvic acid is converted to a 2-carbon acetate. So check it out, just for fun. You don't have to know this for the test, but you had 3 carbons going in pyruvate. Here's one of those carbons, goes out as CO2, and then there's 2 carbons left. Okay, so that's the linker step. That'll be in the Krebs cycle. All right, so here's the pyruvate that went in from the end of glycolysis. Here's the acetyl-CoA that's going to go into the Krebs cycle. Most ATP is not made in glycolysis, and most ATP is not made in the Krebs cycle either. Most is made in the electron transport chain, which I've been telling you, you make a buttload of ATP if you can get your electrons to the electron transport chain. And the electron transport chain is going to use this process called oxidative phosphorylation of ATP. So you're going to take ADP with a D and phosphorylate it with ATP in an oxidative process, and also via this process called chemiosmosis that we'll look at in a minute. But if you're acetyl-CoA, you get in here to the Krebs cycle, so let's check it out. You get this 4-carbon molecule comes in as oxaloacetic acid. You've got a 2-carbon acetate and acetyl-CoA, and then you link them together, and you get 1, 2, 3, 4, 5, 6 carbon, a 6-carbon citric acid. Then you start, and so sometimes they call the Krebs cycle the citric acid cycle. That's why. So anyway, then you get your citric acid. Doesn't show the enzymes again. There's no enzymes shown here, but you have an enzyme. Take citric acid to isocitric acid, and then isocitric to alpha-ketogluteric acid. But look what happens right here. You get an NADH, and that can go to the electron transport chain. You also get some carbon dioxide, which you breathe out. Check it out. You get another NADH here as you go around the Krebs cycle. Oh, woohoo! Every pyruvic acid molecule that gets converted to acetyl-CoA and goes into Krebs, you get an ATP. And then now this is new. This is a new guy, FADH2. So you're going to put in FAD and make FADH2. This also is going to carry electrons to the electron transport chain. And then here you get another NADH, which is going to go to the electron transport chain. So basically the Krebs cycle for one glucose that through glycolysis became, excuse me, 2 pyruvic acids, and then those became 2 acetyl-CoAs, or 2 carbon acetate molecules. For every one of those, you're going to get 1 ATP. So for a glucose molecule, you'll get 2 ATPs from the Krebs cycle. Not that many. But you do get all these electron carriers, NADH and FADH2, and you get to take those to the electron transport chain where you make a buttload of ATP. And here's a cartoon of the electron transport chain. So here's our hero in microbiology. Here's a bacterium, and they cut out this little square here, and they show here's the ECF. Here is the intracellular fluid, or ICF, and then here is the cytoplasmic membrane. There's the cell wall. And then they also show a mitochondria. This would be, so this, you wouldn't have this in a prokaryote, which is bacterium's prokaryote. This would be in eukaryote. So this is what's happening in our cells, in our mitochondria. This process in the electron transport chain is happening here in the intramembrane space in the matrix. So this is a phospholipid bilayer. So this would either be the cell membrane of the bacteria, or in our cells, it would be the cell membrane of the mitochondria. And you don't have to know all the names of these guys, but these guys, so FMN, ubiquinone, cytochrome B, cytochrome C, cytochrome A, these guys are all going to do redox reactions. So they're going to accept electrons and release electrons, accept electrons and release electrons. And here's our heroes, NADH and FADH2 coming, NADH coming from glycolysis, the linker step, and the Krebs cycle, and FADH2 coming from the Krebs cycle, are going to bring their electrons to the electron transport chain. And then through really cool biochemistry, those electrons are going to be transported, and you can see the electrons here, E minus. Those electrons are going to be transported down this chain. When the electrons are transported, energy is released, and hydrogen ion, H plus, is pumped from the cytoplasm of a prokaryote, or the matrix of a mitochondria, into the extracellular fluid, or excuse me, the space, if we're in a bacteria, this would be the phospholipid bilayer, and then the cell wall would be out here. So those free hydrogen ions are pumped in this space between the cell wall and the phospholipid bilayer. And then in our cells, in eukaryotes, the free hydrogen would be pumped into the inner membrane space. Why do you want to pump free hydrogens? Using the energy of electrons, why do you want to pump the free hydrogens into this space? That's because if you build up a really high concentration of hydrogen ions here, you'll build a high concentration here and a low concentration inside the cell. That means you have a gradient, a hydrogen ion gradient, and when that happens and this ATP synthase is open, those free hydrogens will move into the ATP synthase, and when that happens, a whole bunch of magical biochemistry occurs, and ADP gets phosphorylated to ATP. And remember, in the electron transport chain, I said you could make 32, 34, 36 ATPs, but it depends on the cell, but you can make a buttload of ATPs with this process in the electron transport chain. So let's look at the bullets. Energy released from electron transport down the electron transport chain is used to pump protons across a membrane. So H plus cation is also called a proton because it doesn't have an electron on it, because it gave its electron to the electron transport chain. This occurs in the cell membrane in prokaryotes like bacteria, and the inner mitochondrial membrane in eukaryotes. And what I just said is you establish this hydrogen ion or proton gradient, and then when these protons or hydrogens flow through this thing called an ATP synthase, and they call it this, when the hydrogen or the protons move through the ATP synthase, they call that the proton motive force and chemiosmosis. So it's osmosis of protons because they're osmotically moving down their gradient. And then you get ATP synthase to do this process, oxidative phosphorylation of ADP to ATP. So ATP synthase, that's an enzyme, so it's going to take ADP and put a phosphate on ADP, and you make ATP. And if you did a search for ATP synthase, there's like thousands of scientific papers written on the ATP synthase. Okay, So that was glycolysis, the linker step, Krebs cycle, and electron transport if oxygen is present. But what if oxygen is not present in the cell? That means you have to do this process called fermentation. So the purpose of fermentation is to recycle NADH back to NAD+. So in fermentation, NADH is going to come over here. It's going to donate its electron in the form of a hydrogen ion. It's going to, and then when the hydrogen leaves NADH, you're going to make more NAD+. NAD+ is going to come back here to glycolysis, and you can keep glycolysis going. That means you can keep making ATP. So if there's no oxygen or other final electron acceptor available, oh yeah, I forgot to tell you that at the end of electron transport, there's a free electron, and something has to accept it because you don't want free electrons zipping around in your cells. Oxygen, the oxygen molecule, O2, is the final electron acceptor in the electron transport chain when there's oxygen present. So you can have other final electron acceptors. Our cells use oxygen as a final electron acceptor in the electron transport, but you can use other final electron acceptors. If there's no molecular oxygen present, ATP could be made in glycolysis, but it needs a continuous supply of NAD+. So if you don't have NAD+ coming at this point in the reaction series, glycolysis stops. You can't do glycolysis anymore. So if you can make a process, which Mother Nature did, where NADH gives its electron and hydrogen to molecules in fermentation process, then NAD+ can be regenerated, and you can just keep doing this cycle over and over again. Oops, I wrote red on that. There we go. In a bacterial cell, glycolysis precedes fermentation, which glycolysis uses one glucose molecule to produce a net gain of 2 ATP molecules. The purpose then of fermentation, when no oxygen is present, is to recycle NADH to NAD+. So you can keep doing glycolysis and fermentation. You can keep making ATP so you don't die. All right, so if you can keep recycling NAD+ and NADH, then you can continue to make the 2 pyruvic acids. Those pyruvic acids can come over here. So this is fermentation. And you can make carbon dioxide. You can recycle NADH to NAD+. That NAD+ can go back over here to glycolysis. But the problem is you make these byproducts, or lactic acid. That's what we make in our muscles when we're running out of, we're working so hard we can't get enough oxygen to our muscle cells. And some other cells will take that pyruvic acid and make acetaldehyde, which will then be converted to ethanol, which is alcohol. And let's look at the bullet. If oxygen is not available, some cells use this fermentation. It says to partially oxidize sugar. So remember, the sugar they're talking about is glucose that came in here. So it gets partially oxidized or broken down to release energy. But in fermentation, you use an organic molecule from within the cell as a final electron acceptor. Remember, if oxygen is present, oxygen can be the final electron acceptor in the electron transport chain. But if you can't go to the electron transport chain, you can use an organic molecule to be the final electron acceptor. This process of fermentation reoxidizes NADH. So to NAD+, while at the same time it reduces organic molecules. So like pyruvic acid via these common pathways, lactic acid or alcohol pathway. A continual supply of NAD plus is critical to do glycolysis. If you don't do glycolysis, you can't make ATP and you die. So you have to, Mother Nature had to devise a way to make a continual supply of NAD plus over here in glycolysis. And this is the way she did it with fermentation. So if you are a cell and oxygen is not present, you can keep making 2 ATP per cycle of glycolysis if you use fermentation. There's hundreds of possible fermentation end products. We just show here ethanol and lactic acid, but it depends on the cell. You can make a whole bunch of fermentation end products. Fermentation produces a wide variety of organic wastes. They're wastes to the cells that make them, but potentially they're useful to us. Okay. So oftentimes fermentation will make an acid, and those acids could be acetic acid. That's a common name for vinegar. And then pH, you can get a pH change if you put an acidic solution into an acid indicator, it will change color. So basically the 3 chemical products of fermentation are, you can make an acid, you can make an alcohol, or you can make a gas. And let's check it out. So here's this cartoon. It shows glucose goes in, NAD plus it gets recycled, or NADH gets recycled to NAD plus in fermentation. So you can keep doing glycolysis and fermentation. But look at all these things you can make. So Propionibacterium, using this process of fermentation, gives off CO2 and propionic acid. And look what the product is. Delicious Swiss cheese. These organisms make cheddar cheese, yogurt and soy sauce. Saccharomyces gives off CO2, bubbles, makes ethanol, makes wine and beer. Clostridium makes nail polish remover. And Escherichia acetobacter makes vinegar. Laboratory tests look for fermentation products to ID microbes. So we're going to do that in the lab. This is just a table. It compares aerobic respiration, anaerobic respiration, and fermentation. So in aerobic respiration, is oxygen required? Yes. Types of phosphorylation: substrate level phosphorylation and oxidative phosphorylation. Substrate level phosphorylation is just when you that happens in glycolysis, when you take a substrate and phosphorylate ADP to ATP. Oxidative phosphorylation, that's when you use the ATP synthase to take ADP to ATP. Final electron acceptor in aerobic respiration is oxygen. And the potential ATP you can produce in a prokaryote, you can make 38 ATP. You can make 36 in our cells. Anaerobic respiration and fermentation, oxygen is not required. Anaerobic respiration, you can phosphorylate ADP to ATP via substrate level and oxidative phosphorylation. But in fermentation, you only do substrate level phosphorylation. In anaerobic respiration, the final electron acceptor, look at all these final electron acceptors. I think this is nitrite or nitrate. This is sulfite or sulfate. I'm not sure what that one is. CO3. I mean, CO2 obviously is carbon dioxide. And then you can make up to 36 ATP with this. In fermentation, just whatever cellular organic molecule is there, you can use as your final electron acceptor. And I think that's the end.